Hi, everyone. Welcome to Genetics in Your World. I'm your host, Daniel Baker from the University of Pennsylvania. And today we have a really special episode where we sit down with the world-renowned geneticist, Dr. Francis Collins. Francis has had a truly storied career making seminal contributions to human genetics, leading the Human Genome Project, and serving 3 different presidents as the director of the NIH. In this episode, we delve into his initial pathway, learn about how he navigated some early failures, and some really interesting stories about how he decided where to start up his first lab. There's a lot of great advice throughout the entire episode, and if you stick around to the very end, you'll hear Francis's take on the areas of science that excite him the most today. So without any further delay, please enjoy my conversation with Dr. Francis Collins. So maybe to get started, it would be great to hear a little bit more about those early years. And I'm particularly interested to know how, a boy from small town Virginia ended up getting a PhD in physical chemistry.
How did that happen? No one would have predicted it. I grew up on a small farm that had no indoor plumbing and was homeschooled by my parents until the 6th grade, not for religious reasons, but just because they thought they were better educators than the local county schools, and they might have been right. But they were not scientists. My dad was a drama professor. My mother was a playwright. Music was a big thing in our household, and community theater was a big thing. Science, not so much. When I got to be 10 years old, we moved in town to my grandmother's place because she'd had a stroke and needed somebody to be in the house. And my parents decided, okay, we'll let him go to public school now because their schools are a little better. And they were probably kind of worn out because I'm the youngest of 4 boys. And so there's been a lot of homeschooling by then. And it was great because they were good schools. And in the 10th grade, I took a class in chemistry that transformed me because it was taught brilliantly by somebody who helped me understand that science is really like a detective story. You have things that you use in terms of tools and you follow clues. And if all goes well, you learn something about how the universe is put together that nobody knew before. And what could be more awesome than that? Mm-hmm. So I figured, okay, I have found my North Star. I must be intended to be a chemist. By the way, I took a biology course. Oh, it was all descriptive. It was messy. It was not intellectually interesting at all. So I said, life science, no, not going to do that. So I went to UVA, University of Virginia, and majored in, guess what, chemistry. Took every chemistry and physics and math course that I could and ignored most everything else. And then went off to get a PhD at Yale in physical chemistry, particularly in quantum mechanics, which I found absolutely incredibly exciting. And my PhD, which was partly pencil and paper trying to solve second-order differential equations, and partly doing computer coding, which in those days was on cards that you carried around in big boxes. And it was fun, and I kind of got a bang out of it. But, I kind of looked down the pathway of what might be my future, and it didn't necessarily seem all that sustaining. And somewhere along the way, talking to other graduate students in this department of chemistry, some of whom were working on the chemistry of DNA, I realized I might have missed something here. Life science actually does have organizing principles. It does make sense. It has an information molecule. That's really calling to me. So I had a big crisis of my sort of personal vision and decided I'd better pay attention to that. And if there was going to be a change in the pathway, it had better be then. And if I was going to actually step away from chemistry and into life science, I wanted to step all the way into something that might even involve interactions with people who were sick. So despite having had really not the slightest inclination of interest in medicine for my first 20 years, in my 21st year, I decided to apply to medical school. And the good folks at University of North Carolina, for some reason, decided it was worth taking a chance. So you can see there's a theme here. I have the most nonlinear of pathways when it comes to career decisions. It just seems to bounce around randomly, but it all kind of works out eventually.
At that point, when you were transitioning to medicine, were you open to the idea that maybe all you would do is treat patients, or was at that point, were you sure that research is going to play some role in this career that I'm doing?
Yeah, no, good question. I was sufficiently shaken up about having gone this far to getting a PhD and then deciding it wasn't what I wanted to do, that maybe I didn't want to do research at all. And medicine kind of kept a lot of options open. So maybe I'd just be a doc somewhere.
Okay. And so at what stage during medical school did you, or maybe it was later, did you get really convinced that maybe this research thing is the thing I want to pursue?
It didn't take very long. It was like the third month, I think, of the first year of medical school. And what did it for me was a 2-week course taught by a pediatrician about genetics, about human genetics, about medical genetics. Because here was this combination of what I'd been longing for, something that had to do with people, but it also tapped into this digital part of my brain that loved the idea of being able to understand something on the basis of a code, in this case, the DNA code. The physician brought patients to class, and we were all pretty hungry for that the third month of just being in lectures and reading textbooks. So he brought a patient with sickle cell disease. He brought a patient with neurofibromatosis. He brought a child with Down syndrome. And those were just riveting, particularly the ones like sickle cell. Like one misspelled letter out of the whole genome is enough to do this to that person who suffered terribly from sickle cell crises his whole life. Boy, I'd love to figure out how to do something about that. So I think at that point, I pretty much decided, yes, I do want to do research. I want to try to figure out how this can help people and not just be a means of writing about it. And I thought maybe I ought to be a pediatrician, but when I got to the clinical part of medicine, I loved internal medicine more. And I was a little worried about that because people around me like, wait a minute, you're going to be an internist and you're going to do genetics? There's nothing there. I guess they hadn't met Victor McCusick,
Sure,
who I did at some point get to meet, and reassured me that there was actually a lot of promise in this field.
So at what stage did you decide? I mean, I know you did a residency and then you also went and did a postdoc. How did you think about those decisions?
I think I pretty much got in my head that I wanted to be a really good doc. I wanted to get fully trained as a clinician, but I also wanted to move my pathway forward so that I'd have this sort of triple experience that one wanted to aim for, and I hope still do, which is that you would take care of patients and you'd be good at that, and you'd do research and you'd have a lab that was really doing interesting stuff and not just derivative questions. And you teach, because I enjoy the experience of sharing my excitement about things with others, including medical students. So that was the sort of path I wanted to be on. It took a while to get there. I mean, the clinical training to be an internist is pretty much all consuming. So I did my 4 years of medical school, then I did 3 years of residency, and I even stayed an extra year to be chief resident at the North Carolina Memorial Hospital. By the way, I learned a lot there about both medicine and also about how to build teams and how to manage complicated situations and sometimes complicated people, which is, I think, served me well and down the road with other team efforts that weren't always so easy. But then, I got out of that clinical phase, and then I landed in this postdoc to learn molecular genetics. And this was at Yale with the remarkable mentor of Sherman Weissman. And it was really excruciating to have moved from a place where I was a pretty darn good doc. And, you couldn't throw too many things at me in the wards that I wouldn't have a pretty good idea what to do. And now I'm suddenly in a lab where everybody in the lab is just like this, well-tuned machine about designing and doing experiments. And I'm like, how does that autoclave work? Because I'd never done molecular biology. Everybody said, oh, you're a PhD, you should know this. Well, yeah, if it was, some Fortran cards maybe, but you guys aren't really depending on that so much. So the first 6 months as a postdoc, I almost quit. My first project turned out to be a disaster. And I should have probably figured out sooner than I did that it wasn't going to work. So I spent a lot of time on what was going to be my contribution. And at the end of 6 months, I had basically nothing to show for it. And I felt the clock ticking. And I had a mentor who was both the smartest guy that I have ever had the chance to work with, Sherman Weissman, and one of the worst communicators. So half the time, I didn't know what he was talking about. And I couldn't tell when he sort of bubbled up with some idea, oh, am I supposed to do something with that? Or is this just for fun? And it was never quite clear which one it was. I was greatly blessed by then having him introduce me to a colleague, Bernie Forget, and then to be assigned a project in hemoglobinopathies, particularly on hereditary persistence of fetal hemoglobin. And there was something concrete, and Sherman's incredible brilliance, but being off in the clouds, was nicely balanced by Bernie, who was very clear, very transparent about what needed to be done, and a wonderfully warm person. And it all finally started to come together. And then, you have that moment where, it's 1982, and I've been laboring on this effort for a very important cloning project to try to understand the molecular basis of how fetal hemoglobin stays on in these families that have that condition. And I've got the clones, and I'm turning on the switch in the dark room on the ultraviolet to see what's happened with my latest restriction digest, and bang, there it is. Like, okay, I want to do this forever.
Yeah.
I think that's a good message. When you first are starting out, you probably have this experience already, Daniel. Your experiments become very much sort of part of your being, and an experiment that fails feels like you failed. Even if you did everything right, it just turned out the hypothesis was wrong. It still feels like, oh, I'm not meant to be here. You have to get through that. And we all do get through that. Whatever you're working on, if it doesn't fail most of the time, it's probably not an interesting project. Right? And so failure is part of it, but you do try to learn from it. And then once in a while, something works, and that keeps you going. And
yeah. So obviously your trajectory, it sounds like you are a person who succeeded all the time. So really hitting your head against something. How did you deal with failure? Maybe take us into some of those moments. What was it like, and what did you do?
When the first failures really hit me hard, and like I said, I almost thought this is a sign that I am not supposed to be doing this. And maybe that idea of just being a doc, because I think I know how to do that, is really what my calling is. And I considered very much making that speech to my mentor and packing my stuff up. But it didn't feel quite right. It felt a little premature. It felt like, okay, you got to give this a little bit more time before you draw that sweeping conclusion. So yes, sticking to it, being persistent. And certainly throughout my whole career, I would say most of the experimental things that I've been involved in have failed a lot more than they have succeeded. But again, as I said, I hope I'm now at the point where when something fails, it's not like, I never want to think about that again. It's like, okay, what was it about that failure that actually has some useful information to help me design the next experiment? And then let's go on and do that.
So as you moved on and began your own lab, I'm curious how you incorporated good things and bad things from previous mentors. I think you already referenced that there were things about Sherm that were fantastic.
Yeah.
And how did you choose to incorporate that with your own trainees and your own students? And how were you able to delineate areas of things that needed improvement for yourself?
Yeah. One thing I learned from Sherman is that if you're going to spend your time in research, you may as well pick a project that's actually significant. And that isn't necessarily the default, especially when you're first starting out and you want to be sure that something works. It's kind of tempting to look at an area that's already pretty well mined and say, well, there's this little piece over here that hasn't been settled yet. And I could design an experiment that almost certainly is going to work. And I can, make some results, and I can probably write a paper. And maybe that's the way to get started. And some people end up kind of doing that for the next several years. But, you don't have that much time in this life. You've only given so many years to be a productive researcher. Maybe it's a good idea to pick something really hard, even if it's got some risk. And then I kind of always felt like have a little bread and butter thing on the side that is more derivative and incremental, where you're pretty sure you're going to make progress, and run both of those, but put your heart and soul into the one that's really hard. And for me, getting to Michigan in 1984, setting up my own lab, having developed a very fledgling idea about how you might go across the whole genome and find a mutation that causes a disease that we don't understand. I decided, okay, I'm going after cystic fibrosis, which was a complete black box at that point. And boy, there were so many failures in that one. It took a full 5 years. It burned out an awful lot of people's time with things that turned out to be dead ends, and ultimately succeeded. Here's another lesson: by figuring out how to merge my lab with our main competitor, Lab Chi Choi's lab in Toronto, because he had a particular approach and I had an approach that was complementary. And putting them together and putting our teams together so they all became friends and colleagues just made a huge difference. We were able to move, 5 times faster as we would have been able to if we were just competing. I've never forgotten that. So it got there, but it was 5 years and it was very high risk of just coming up with nothing. Most people, it's hard to believe right now, most people in the mid 1980s really thought it was not going to be possible with the genome being so big to find the cause of a disease that might just have 1 or 2 or 3 letters out of place, unless you had something like a knowledge of what the protein product was, which we did not have, or maybe a cytogenetic rearrangement, a breakpoint that you could kind of focus on. That's how we got muscular dystrophy, for instance. Cystic fibrosis didn't offer any of that. And it kind of then became the proof of principle that it is possible to get the answer. But boy, it was hard. And boy, did we need a genome in order to get there ever again for any other disease. So that became a major motivator for me and for everybody else to do the genome project.
So I just want to jump back just for a quick second. It sounds like the environment at Michigan was really important. And I am curious as to how you zeroed in on that as you were trying to make your decision of where to set up shop.
It was tough. I had been pretty happy at Yale. There was an indication that I could be offered a faculty position at Yale to stay there, and that would have been the easy default. I also looked at a position at Penn back in the day when Penn was building its human genetics, and a position in Seattle. And then Michigan sort of turned up. And the head of the Division of Medical Genetics was just after me. He was on the phone. He was sending me letters, said, you got to come and look at this. And I was like, okay, I guess I should just go and look. It's sort of part of the thoroughness of the search. And what did it for me really was the way in which the chairman of medicine at Michigan, Bill Kelly, had organized the effort there to be just maximally supportive for physician scientists. He was recruiting a whole cohort of people at the same sort of level that I was, who then became some of my best buddies and are friends for life. People like David Ginsburg or Craig Thompson, who just stepped down as the head of Sloan Kettering, or Jim Wilson, who was obviously a little controversial, but has done amazing science. Yeah. You know him well there at Penn. Or Jeff Leiden, who went on to become the CEO of Vertex. All of those people were just like down the hall. And you had this amazing opportunity then to try out ideas on each other. And Kelly made it possible for you to have protected time for research, although he wanted you to do your 1 month attending, and we all wanted to do that. But he would also arrange it so if you got grant funding, he had a way of kind of putting some of those funds in a research area that you could use for a new idea, and not too many places would do that. So it was just the perfect environment. The space was great, the collaborations were great, and the tightness between the bench and the bedside was just what I wanted to see. And none of the other places really offered that. So I think somewhat to the shock of people like Lee Rosenberg, who at the time was the chair at Yale, I said, no, I'm not going to stay here. I'm going to Michigan. He's like, oh, biggest mistake of your life. He later admitted that he probably underestimated what a great place it was going to be for me.
So it sounds like collaboration, both internally and externally, has been really key to your career. As you've been thinking about the Human Genome Project, I mean, how important were those integral experiences of learning to, talk to your own colleagues as well as competitors play a role as you were trying to do this kind of like worldwide national experience?
Oh, I think it was critical to have learned how much you can gain by collaborative relationships, and not just something that's rigid and legalistic, but something where you really learn to know each other, to trust each other, to become friends, to enjoy the experience of brainstorming when you don't really quite know where it's going. So you become undefended in terms of your own ideas.
Mhm.
That happened for me with cystic fibrosis. When the Genome Project got started, it was pretty clear we had to have a lot of that if it was going to work. I did not expect to be asked to come and lead that. And that happened after the resignation of Jim Watson for one of the moments where he said some things he shouldn't have and was suddenly sent packing. And then here I was supposed to organize this bold effort. And this was the first really big science effort in biology. Sure, that had been happening in physics, but in biology, there really wasn't anything like this. And a lot of people were skeptical, well, maybe even more than skeptical. They were trying to kill the project.
Mm.
And no way it was going to work unless we really pulled together a team who would share this vision and would be willing to drop everything else that they were going to do to work on this historic effort to read out our own instruction book. So a lot of what I had to do was recruiting, and I had to convince people that this was something so much better than anything else they might have spent their time on. It kind of worked. And as the main success of the Genome Project really was building that critical mass of really brilliant people, and then convincing the Congress to provide some resources so that we could go forward. And all the rest of it just kind of clicked. It took some management. There were times where, yeah, somebody's ego got a little out of control. And we had these meetings every Friday at 11 o'clock of the principal investigators of the centers that were doing the largest amount of sequencing. And I always tried to turn those into lab meetings, but then it got to the point where you sort of had to review progress. And occasionally somebody would get a little out of line, and I'd have to take them to the woodshed afterward and say, that's really not the way you should talk to your colleagues. Because that was important. You don't want that kind of friction to get out of control. And it all worked. And, ultimately 2,400 scientists, 6 countries, all putting their shoulder to the same wheel, recognizing you can't say you did the genome if part of one chromosome is a mess. It's all got to get done. It worked, and nobody worried too much about who was going to get the credit because it was so significant. It mattered. It was going to make a difference for decades, centuries to come.
Yeah, one of the things I'm fascinated by, both when you were the director of the Human Genome Project and ultimately the director of the NIH, is you've always had a vested interest in continuing the work of your lab.
Yes.
And I would love to know, what's the impetus behind that?
It's 2 things. One of it's mental health. Because if you get into this administrative role and dealing with the Congress and worrying about budgets, it can kind of grind you down, and you maybe start to forget about really what matters, which is getting the science done. So part of it is mental health, for the ability to kind of keep myself grounded. And the other part of it really was a dream that I had of not just being an observer, but a player of some sort in applying all of these transformative efforts to try to do something about diseases that are still causing a lot of human suffering. And for me, that has been a variety of diseases over time. Right now, it's all about type 2 diabetes on the one hand, and a rare form of premature aging called progeria on the other. 2 projects that are very different in terms of the kind of science that they're currently utilizing. But with the 10 people in my lab, we just have a wonderful time tapping into every new observation, every new development, and seeing how we can apply it to those conditions.
Okay. So maybe just 2 last questions. The first is, I mean, if I could take you back to when you were gene hunting and finding the genes for NF or progeria, what did you hope would come of discovering what the mistakes were? Did you ever think there would come a day when we could fix those genes precisely the way we maybe can do?
I did not think there would be a day where you could actually go in and fix the mutation, as we now are doing with CRISPR and prime editing. I did dream that just knowing the mutation ought to lead you to some kind of insight that would provide an intervention that would be better than what we have. And take cystic fibrosis, for example. When we first found the Delta F508 mutation in 1989, which 90% of CF patients have one or two copies of that, I thought, this is a great candidate for gene therapy. This is a gene that's expressed in the airway. That's the main place you want to treat. So let's just stitch a normal copy of CFTR into some vector. Oh, an adenovirus sounds good. And put it into an aerosol and bang, everybody will get better. And as you know, that basically got us nowhere. I should have had better education in immunology. The immune system was going to have none of this. What are you putting into my airway? I don't think so. And so we spent several years really banging away at that before it was pretty clear there had to be some other strategy. And then the small molecule effort arose, particularly because the Cystic Fibrosis Foundation was willing to take a big risk and put a big bet on that, which a lot of people were skeptical of. But look where we are now, Daniel. I mean, gosh, Trikafta, the stories that I hear almost every week from people with CF who are in bad shape, maybe on the transplant list, many of them certainly having figured out that they ought to get their affairs in order. And on Trikafta, some of them, back at work and thinking about, well, maybe I'll try this and maybe I'll try that. And someday I'll retire like everybody else. It's just astounding. It's miraculous to see what happened. Now, it took 30 years of incredible hard work. And I don't want that, which is why I'm so excited about the idea of doing gene editing. Because gene editing doesn't require you to understand the intricacies of the molecular biology and the chemistry of every gene defect. You could scale gene editing. Once you figure out you've got the right delivery systems and you've got the right apparatus to search and fix, find and replace the misspelling, then there's thousands, probably 6,500 genetic diseases that are waiting for that application to happen. And I don't think it's crazy to aim to get to all of those in the next 10 or 20 years.
And then my last question for you is this. If I could take you back to when you were a resident and a postdoc thinking about the big questions that you were going to tackle in your career, if you had to start over today with so much changing, what are the fields and the questions that excite you the most today?
Well, I was certainly thinking about genetic disorders and what we might do, and not just to find their cause, although that would be a good start, but actually offer ways to alleviate suffering and prolong life. And that pretty much has sustained me ever since. And it's not just me, it's the whole community now making this amazing set of advances happen. I wish I'd paid more attention, maybe in a few other places that are now just really exciting. Neuroscience, this is the period of time where we're going to figure out how the brain actually works after many decades of describing it. But now, we just have this cell census of the human brain, all those cell types that you can analyze with single cell RNA-seq and ATAC-seq, and you can really see what they're doing. And it won't be long before that adds up into circuit understandings that tell us things like how you lay down a memory and how you retrieve it. And we certainly know that so much of the morbidity and mortality in our species is neurological, neurodegenerative disorders, and we finally will have a foundation to begin to understand those. So that's a place, if I was starting over right now, I might decide to be a neuroscientist. I wish I had maybe along the way done more to nurture my computational capabilities. That's what I did as a quantum mechanics PhD, but I haven't done a whole lot since. And now it seems that computational and artificial intelligence approaches are just taking off. In fact, my own lab now has more people doing that than working at the bench. But I feel a little bit less well-informed than I would like to be about the opportunities and the limitations in that space. And then immunology, again, I once made a joke when I was searching for a gene that would be responsible for some hereditary disease. Please, dear God, don't let it be a gene in the immune system, because I don't know what to do with that. And that was unfortunate because, well, Daniel, the immune system is like this incredible engine of opportunity. And I would have done better, I think, to pay more attention to how it works.
Well, I'm glad you chose what you did choose. And thanks for taking the time. I really appreciate it.
Well, it's good to talk to you. I hope anybody listening to this finds it encouraging and not just confusing. But the last thing I would say for trainees who are getting into this field right now, you have arrived at exactly the right time. We are on this exponential curve of knowledge acquisition, and it's going to go rocketing upward for a few decades to come here. And to be part of that, to be contributing to that and be part of what's happening all around you is just going to be exhilarating. Yeah, it's hard work. Yeah, there's this concern about funding. Work-life balance has to be attended to. Do, as you get the chance, figure out how to work in teams that can go faster, because you'll find it really satisfying to see the progress, and you'll make great friends along the way. And don't worry, you'll get the credit if you're part of the team and you've done something substantive. It's not going to dilute you. It's going to enhance your reputation and your skills. Science is a social activity, and it should be. It's more fun that way.
Mm-hmm. Well, thanks. Thanks so much, and look forward to chatting with you sometime in the future.
I look forward to that too, Daniel. Thanks a lot. Thanks for doing this.
Special thanks for listening to this episode of Genetics in Your World. This podcast is produced by the Early Career Leadership Program of the Genetic Society of America. Thanks for making the time, and until next time,